Hello, student. With the continuation with the previous video, that is step by step molecular docking. Today we are going to see the preparation of protein or macromolecule for molecular docking. For that, we are going to open again Discovery Studio and Autodock tool. Okay, now this Discovery Studio visualizer is now open, but still some time is required to open Autodock. Okay, so today we are going to take one example of protein preparation. So here I am going to take a example as a glycogen synthase kinase, which is an enzyme required in the metabolism of glucose. So let us download the protein from PDB protein data bank. So we are going to type PDB in Google. So PDB database. Okay, so here it is showing that RCSB PDB homepage. So this is a huge database of protein crystals available. So proteins with a different codes, there are more than 100 proteins may be available for a single enzyme, but these proteins are complex with various cofactor or various ions, or you can say that various native ligands, but depending upon our requirement, we are going to select them. So here I am going to type glycogen synthase kinase. Okay, so here it is showing that glycogen synthase kinase 3. So click on this. So here you can take your interest of a protein on which you are going to doing your research. Okay, now this protein data bank is showing the information with this glycogen synthase kinase bound with this AZD. This is a one ligand available. So we will check on next page. So here it is, you can take this anyone, for example. So this code you can find from literature also in research paper. The code of a particular protein is mentioned on which molecular docking is carried out. So you can take that one, or you can take any one of the protein. So for selection of protein, there are a number of parameters are required. So that parameters you can compare. So here the experimental data snapshot is mentioned, which gives that X-ray diffraction was the technique which is used, and the resolution of these protein were found to be 1.74 angstrom. Then R value free, that is a 0.2, R value work, and this is an observed value. So their difference between the R free value and R observed value, that must be minimum, that must be the close. So this is a requirement. And also there is a one more requirement, that is a residue count. So here the residue count is showing that a number of amino acid present in this protein is 351, but if you know the standard amino acid present on a particular protein is get varied from what the amino acid number is showing over here, it means that a difference between the mention or actual and here it is available, that state that number of amino acid is missing. So that residues gives or disturb the geometry or quality. So here the quality assessment was done, which showing that's a worse and better. So it is showing that this showing a better one. Again, one more technique is there to check the quality of the protein. It's called as the study of Ramachandran plot. So here we will have a look on a Ramachandran plot study. But before proceeding to the Ramachandran study, let us see certain data mentioned in this PDB. So this is a refinement. These all values indicating certain qualities of this protein. So this R factor is important while deciding the protein. So difference between R value and R observed, or R free and R work, that must be the minimum. Then the residue count, that must be a very close to the residue count which must be available for that protein. So if it is not available, then simple, you have to go for another protein. So here I know that 6V6L is a protein of glycogen synthase kinase that I studied before. So I am going to show you the parameters for these 6V6L. So now what they mention, that is a co-structure of a human glycogen synthase kinase beta with this is a native ligand. So it is get co-crystallized with this native ligand. Okay. So here again some information is mentioned. So the organism is a Homo sapien, from where it is being collected. Then these crystal is deposited here in 2019. So while selecting, you must be aware that the co-crystal must not be a much older. So there must be the recent one. So now look at this resolution is a 2.19 Armstrong, means it's showing a good resolution value. Then difference between R free, R work and R observed is considerable in between. So R work and observed, so it is a 0.001 difference is there. So it is quite good for our selection. But apart from that, the one more advantage of using RCSB PDB is that they mention at least one literature in which these your selected protein code for a particular protein is mentioned, is given. So discovery and optimization of novel pyridine as a highly potent selective glycogen synthase kinase 3 beta inhibitor. So you can open it and you can see this, that what kind of parameters they use with this and what the result they got while using this protein code. So as I told you, so there is one study assessment for to check the quality of the protein is called as Ramachandran plot. So we can click on Ram plot, or you can Ramachandran plot. Just open it. So there is one more application is there to check the Ramachandran plot. So this is a Ramachandran plot you may find over here. If you put Ramachandran plot server, then it will appear in the Google. But there is one more website is to check RAM plot called as a ProCheck. ProCheck is to study the quality of the protein. Okay, so now ProCheck is available over here. So ProCheck is available here, download from here. So you have to click on this PDB sum. So click on PDB sum. So it will redirect on PDB sum homepage. Okay, so here you have to enter the PDB code that of your protein under your study. So here my protein is a glycogen synthase kinase, but out of this number of glycogen synthase kinase, I want to check the quality of this 6V6L, whether it is good for my study or not. So for that reason, I am going to enter here 6V6L and just click on find. Okay, now there is a project data is available. So it indicates the all information that is in December 19 it was available on the this database, and the release date was December 20. So the same information was matched with the PDB. So you have to click on this project. Okay, so now this is a plot for this 6V protein. So what this plot indicates? So here it is a psi and phi angles. So these psi and phi angles are generally the bond angles between the amide and carbonyl bond. So in a protein there is a carbonyl group and amide bond that makes a particular angle, and that angle were drawn in this plot. And what this angle stated, that if that angle is perfect, if all these dots means these are the protein residues having good angle of psi and phi angle, if they are good, then they must be fall in this red zone. So it is a requirement of the protein that the all amino acid residues from the protein must be fall in this red area. It means that it's showing a good psi and phi angle. So look at this, one dot is outside this yellow area. So it indicates that this particular amino acid is not having good angle, so it can distort the geometry. So this amino acid is responsible to distort the geometry of the protein. So simultaneously this red area is the highest, where the all possible dots or residue must be present. Secondary, this area other outside this red indicating